In today's class, I'm going to talk about something which is very important and technical, plus it's little long as well. So please have patience and listen to it carefully because in last 4 years of acoustics teaching, I have realized that this is one concept where students make create a lot of confusion in their minds and their doubts remain. So I'm going to talk about them. After the online streaming gets over, we will I will make a Google Meet for a short doubt discussions. So if you have doubts, then you join it. Don't join it just for attendance. Attendance I will send you on the WhatsApp group. So you will get it there. Thank you, Nimishka. Also, do join the group Google Meet after the lecture. If you have any issues with joining the Canvas platform. So if you have any doubts you want to discuss, most welcome. Or if you have anything issues with Canvas, then also you are most welcome. So let's start. A very good afternoon to all of you. In this class, we have already in the process of understanding the structure of inner ear. We will continue with that and we will see how exactly the process of hearing takes place. We are going to look at the oscillations of basilar membrane and its details. So inner ear, as we had shown in the last class, has two components. One is cochlea, the way depends on how you pronounce it. This part, which is the organ of balance, not for our concern right now in the present discussion, we focus on this, which is cochlea. As we have already discussed, the cochlea is something which is coiled long organ which transform pressure variations into coded electrical signals. So this lecture is all about understanding that. We have also already seen that what are the cross sections. So we have a scala vestibuli, scala tympani, we have basilar membrane, on that we have the organ of Corti, on which we have hair cells. So as the oscillations takes place, the organ of Corti oscillates here and that sends impulse to the hair cells. Hair cells almost work digitally by using suitable ion exchanges to create a suitable 1 and 0 and sending a voltage signal through the neurons to the brain. So this is all we have already discussed. Let's get into the more detail today. So here is a model of cochlea. In the earlier diagram, I had shown you that it is a helical curled structure. But here now, if you open that curled structure and imagine it to be a straightened out structure, then this is roughly around 35 millimeter in length or 3.5 centimeter. This first hand model has the upper part and the lower part. It has a tapering structure. All the parts are thicker at this end, and as they go down this side they get thinner. The basilar membrane or the central membrane stops just before the end. So it's a tapered cylinder and there is divided by the basilar membrane. It is filled up by a fluid. More details about the fluid you can read. These are the two windows. The oval window is important because oval window, which is closed by a thin membrane, it is on which the bones in the middle ear go and hammer. So when the middle ear bones go and hammer on this, they send a wave of oscillation in this long membrane, long basilar membrane, and creates ripples in the fluid with which it is filled. The cylinders are round. This window is oval. This is a round window. That is how they are separated. So what happens is, at this end there is a small hole. So this hole enables the fluid from this side to get transmitted to this side, so that here when the hammer goes and hits, the oscillations are set in the upper portion of the basilar membrane. But as it goes further and further, the oscillations are then diffused in the lower section through this hole, and the entire fluid can be sent into oscillations. So that helps us balancing the energy which has been imparted from this side. So this is a typical cross section. So stirrup or the middle bone vibrates against the oval window. It keeps vibrating as the sound is transmitted. Hydraulic pressure waves are transmitted rapidly. So these are the hydraulic pressure waves, depending on how the frequency of which will depend on how fast or slow these bones are oscillating, which in turn depends on the external sound stimulus. And that actually involves the fluid starts vibrating, and that also sends vibration in the basilar membrane. You can see these drawings, they start vibrating up and down. And once that happens, then it has been noted, and please note it down, that higher frequencies, high tones, shriller voices, shriller sounds, remember high frequencies are shriller sounds, they are encoded towards the base of the basilar membrane. They are encoded, and that part of the basilar membrane which is close to the oval window. While where the basilar membrane is narrow and stiff. So a narrow and stiff part of the basilar membrane, which is more sensitive to the higher frequencies. As you go away from the base, the basilar membrane becomes little thicker and little softer. So those ends are responsive to the lower frequency. So if you plot a frequency response curves with different frequencies, then you will find something which is very amazing. You will find that, say, 1600 hertz, which is a higher frequency, on the basilar membrane, it gives a peak somewhere around 15-16 centimeter. But if you have a frequency of 50 hertz, then it is sensed somewhere at around 35 millimeter, 15-16 millimeters, sorry, and this is sensed at around 35 millimeter. So two different frequencies are actually sensed at two different locations on the basilar membrane. So that is a very important point in hearing. This is a frequency separation. As we had discussed in the first or just a few lectures back, that human ear is very good in separating the two frequencies, and this is the first step towards that separation. Higher and lower frequencies are separated at different places. It is like a Fourier spectrum. What this bone is transmitting is a complex of frequencies combination of various frequencies. What basilar membrane is doing is that it is separating them by oscillating at different distances, or sensing them at different distances from the base point. So when the so depending on what frequencies are present, different sections of basilar membrane oscillate, sending ripples and signals to different hair cells in that region. So 1600 hertz will give the signal at a hair cell which is located at 16 millimeter, while a 200 hertz signal will give a signal to a hair cell which is located at around 30 millimeter. So that's how they differentiate. This is a graph of distance from the base plotted on the y-axis and different frequencies which are hertz. So in hertz there are different frequencies. So you can see that as you go to a very low frequency, and remember this is not a linear scale. In fact, if you convert into a logarithm, this graph is a straight line. So it's a logarithmic response. When you are close to a very low frequency, you are at a very high distance, 30 millimeter, even very low, you can go up to 35 millimeter. As your distance from the base decreases, the frequency sensitivity goes on increasing. Very high frequencies are sensed very close to the base, where the cochlea, where the basilar membrane is stiffer. But that is why as you age, the basilar membrane gets more and more stiffer and you lose your audibility to the higher frequencies. So you start losing it from this side. Another important thing that you have to notice, that identical musical intervals in the physical spacing along the basilar membranes are almost equal. So if you take two musical intervals, let's say something around 500 hertz and 1000 hertz, so double of this, in musical terms it's called an octave away. So if it's a double of this, then whatever is the gap difference here in distance on the basilar membrane, if you are between 500, it should be 250. So if you are between 500 and 250, then that distance is again the same. So for every time you double the distance on the frequency scale, the distance on the basilar membrane remain the same. And that is why that could be one of the reasons for the presence of octaves in the music. We will discuss more on that later on. So what is happening is, here basilar membrane is differentiating between different frequencies of mechanical vibrations. I repeat, higher frequencies are sensed towards the base of the basilar membrane, lower frequencies are sensed towards the edge or the far end of the basilar membrane. So basilar membrane is doing the frequency separation. It is also done in the brain as well, and we will discuss that in the next chapter. But in this chapter, we are talking about the first step towards frequency separation. The next step is conversion from the mechanical vibration of basilar membrane into electrical impulses in the auditory nerve, and it is achieved by something called as organ of Corti. When the basilar membrane vibrate, the hairs of the hair cell are bent, and that generates nerve impulses that travel to the brain, that actually triggers on and off the motion of ions, the potassium ions in the hair cells, and by which the suitable electrical impulses are created. They are fairly digital in nature, in a sense that they are either 1 or 0. kind of vibrations. And then, depending on what kind of impulse rate is heard, the brain decodes the frequency and intensity of sound. So I have a beautiful video here which I would like to share with all of you.
To hear, our ears must capture sound, transmit it to the organ of Corti, and translate it into neural impulses to be delivered to the brain. The organ of Corti is located in the cochlea, a spiral, three-chambered, snail-like structure, 10 millimeters wide, within a bony matrix. The organ of Corti extends from the anterior part of the vestibule and coils for about 2.5 turns around a bony pillar, called the modulus. In cross-section, the uppermost chamber is called the Scala Vestibuli. The oval window is situated at the base of this chamber. The lowermost chamber is called the Scala Tympani. At the base of this chamber is where the round window is located. Both the Scala Vestibuli and Scala Tympani contain perilymph. Between the Scala Vestibuli and Scala Tympani is the Scala Media. This houses the organ of Corti, which is referred to as the receptor organ of hearing. The Scala Media is filled with endolymph. The Scala Media includes structures from the tectorial membrane, basilar membrane, and hair cells, which sense the mechanical forces. The hair cells are located between the tectorial and basilar membranes. Approximately 16,000 hair cells are within the cochlea. There are two kinds of hair cells, inner and outer hair cells. 95% of the afferent fibers are from the inner hair cells, the sensory receptors that communicate with neurons from cranial nerve 8. Outer hair cells receive mostly efferent input from the superior olivary complex. The filamentous structures that connect the tips of adjacent stereocilia are known as tip links. These are thought to amplify the forces in the area of the molecular sensors. How does sound enter the cochlea? Compression hits the tympanic membrane, causing the stapes to transfer force to the oval window. The sound travels down the Scala Vestibuli, around the helicotrema, to the Scala Tympani, allowing its fluid perilymph to mix. From there, sound moves to the round window. High frequencies are encoded at the base, and low frequencies at the apex. It is this property that leads to the tonotopic map along the basilar membrane. The manner in which the basilar membrane vibrates in response to sound is the key to understanding cochlear function. The hair cells are located between the tectorial and basilar membranes and are stimulated by the shearing force between the two caused by the pivot point of the two membranes. The pivot point of the basilar membrane becomes displaced. The tectorial membrane moves across the tops of the hair cells, causing the stereocilia to bend. The ionic environment of the compartments plays a critical role in signal transduction. The apical portion of the hair cell is bathed in high potassium solution, and the base of the hair cell is bathed in potassium poor solution. This causes the opening of mechanosensitive channels, allowing potassium to flow into the cell, leading to depolarization. This in turn opens calcium channels at the basal end of the cell, leading to vesicular transmitter release to stimulate the nerve. Because the relative voltage and potassium levels are low at the base of the hair cell, potassium flows out of the cell. This establishes that potassium flow through the cell is used for both depolarization, potassium in at the apex, and repolarization, potassium out at the base of the hair cell.
So you can in this short video you also saw that how complex it is. Basically, the vibrations of the basilar membrane, all we have to remember is vibrations of the basilar membrane leads to the stimulus to the neurons, and that happens through a chemical process, or you can call it a mechanochemical process, the process which is mechanical, which in turn leads to chemical release of ions, which gives the electrical signal or impulses to the brain. So yeah, I mean I put presented the chemical process just for you to keep things in perspective. That part is not mandatory for us to follow further. Look at the mechanical process. So let's focus more on that. And with that, we take up the last topic for today's discussion, and that is the presence of critical bands. The critical bands are largely hypothetical, but they have the correlation, experimental correlations. So if you look at those nerve fibers, they respond only to a certain range of frequencies. And for every nerve fiber, you will have a characteristic frequency. Remember, every nerve fiber is in turn going to have many hair cells which are sensitive to the sound. So their maximum frequency has this kind of frequency response. I I suppose you all know what do we mean by frequency response. On the base, you have the frequencies plotted, while on the y-axis you have amplitudes plotted. So a particular neuron, a particular nerve fiber, say which is sensitive to 1046 hertz, will show a peak at that frequency. But moment the frequencies are changed, you will find that its amplitudes of oscillations will drop. But this drop is not symmetrical. If the frequencies are changed on the higher side, the drop is very sharp, while when the frequencies are changed on the lower side, then the drop is slow. Please remember this, because we will come back to this in we are in the 5th chapter right now. When we get into the 7th chapter again, we will talk about it when we talk about sensation of pitch. So this is how the sensitivity of a given nerve fiber is. These all can be checked present day with a very highly sophisticated experiments. So I'm not getting into it, but if you are interested that how these things are verified experimentally, do find it on the net. So now if you have two frequencies, for example, which are separated, now 523 and 1046, now you can see that it's almost double, perfectly double. These two frequencies are, when one is the double of other, we say that they are an octave apart. Octave in music, the 7 8 step sound. What you call in Hindi is sa re ga ma sur. That is, they are octave apart. So this is one particular frequency, this is another particular frequency. Now these two are sensed at two different locations on basilar membrane, as we have just discussed. So they will have very few, their response curves will not overlap, it will hardly overlap, and there will be very few nerve fibers and hairs which will be stimulated by both the frequencies. However, wait, so since there are hardly any nerve fibers, hair cells which are stimulated by both the frequencies because they are physically separated on the basilar membrane by a large distance, you can make out these two sound notes very independently and simultaneously. You can hear them simultaneously. However, if you bring the two notes closer, you can see 523 and 697. If you bring the two notes closer, then these frequency responses start overlapping to each other, and you can see that this common area shows that there are large number of nerve fibers and subsequently the hair cells which are now sensing both the frequencies, and at that time you will find that some sort of sound merging starts happening. You will find it difficult to sense the two notes independently. When you bring the two frequencies even closer, then a situation comes where the two notes are so close that most of their frequency response curves starts overlapping, and in that case lots of nerve fibers sense both the frequencies and they actually give signal to brain of a complex phenomena, complex frequency. They do not identify each frequencies independently. Do you remember now from this that which experiment or concept we are talking about that you have already studied in your earlier class? I give you 10 second, 15 second to think about it. So yeah, if you think about it carefully, you have studied about formation of beats. Now recall the beats experiment we just did in the revision also. If you have two frequencies which are close, you hear beats. That's what you study all the time. Now just look at it in a different way. When the two frequencies are closed enough, then on your basilar membrane they are sensed at two different positions which are also very close. So all the nerve fibers, all the hair cells will have will be stimulated by both the frequencies, and you won't be able to hear the two frequencies independently, but what you hear is a common tone which is called beats. So we are talking about Beets experiment. In fact, Beets is a beautiful experiment. So let's revert that experiment, think more about it. That's right, Atharva. If you think more about it, then in beat formation, if two frequencies are very close, you hear a common tone. You can't hear them separately. Now listen to me very carefully. As you increase the separation between the two frequencies, you hear more and more beats. Okay, you can try this with two cell phones at home. Just put two different tone generator apps in two cell phones, match the frequencies and hear. You will get it. Very simple to get at home. Now, if you separate the two frequencies, you will hear more number of beats because the frequency of beats depends on how fast or what is the difference. But as you keep increasing the two frequencies, a situation will arrive that the separation between the two frequencies would become enough so that very less neurons will be commonly affected by them. Very less nerve fibers would be commonly affected by them. And when that happens, when this shaded area becomes sufficiently low, then in that case, you can hear the two frequencies independently and you will stop hearing the beats, that is the common frequency. So that is what will happen. So now you started from very close and you separated them to an extent that you can just hear them independently. That experiment can tell you the presence of a band in your oral structure. So if the two frequencies are in the same band, then you can't distinguish between them. If the two frequencies are just out of that same band, if they are in two different bands, then you can very well distinguish between them. So such bands can be imagined on the membrane, basilar membrane. Remember, it's a theoretical construct, but it has experimental background. And such bands are normally called critical bands. Okay, so the critical bands, I'll come back to it, but here is some of the data on the critical band. So for example, if you do that Beets experiment at around 100 hertz, then the critical bandwidth is around 90. That means anywhere between say 50 hertz to 140 hertz, if you play on two frequencies, you won't be able to distinguish. But moment if your one frequency is say 60 hertz, let's say, and if the other frequency is 90 more than that, so 60 plus 90 is 150, so other frequencies beyond 150, 160, 170, then you will be able to hear the two frequencies independently. But if the other frequency is in the same bandwidth, which is whose center is 100, so if one is at 60, another is at 120, you will not be able to hear them independently. So that will cause your one critical band. And the bandwidth in which you won't be able to distinguish, or the range of frequencies in which you won't be able to distinguish between the two frequencies, they are called, it's called critical bandwidth. So at these fixed frequencies, 100, 200, 500, 1000, 2000, 5000, 10,000, these are the critical bandwidths. So for example, at 10,000, your around 10,000, your two frequencies has to be at least separated by 1200 hertz to make them audible clearly, separately. So these are all called critical bands. You can see that the critical bandwidth remains constant in the beginning and then increases. So typical variation in the critical bandwidth is like this. This is the way the critical bandwidth varies with the center frequencies. I recommend all of you to memorize this table. Again, I'm reminding you that these data are statistical average performed over large number of people. You can try this by an experimentation at home. Uh, you may find some results which is close to it. So it's nearly constant at low frequency. It is roughly proportional to frequencies at high frequency. So here it's a straight line, and bandwidths vary. So as I said, there are fluctuations depending on what experiment you are performing. I go back to the slide which I had left. When two pure tones are so close in frequency, and this revises the critical bands, when two pure tones are so close in frequency that there is a considerable overlap in their amplitude envelopes on the basilar membrane, they are said to lie within the same critical band. So one critical band can be imagined as a data collection unit on basilar membrane. Remember, there are no physically bands available. The basilar membrane is one long continuous structure. There are no physical divisions. But we have, by experimentation and by theorization, we have divided into bands. So there are around 24 critical bands. They span the entire audible range from very low frequency to very high frequency, about 1.3 millimeter long. So each critical band's width, remember the entire the entire basilar membrane is around 35 mm. So each critical band is around 1.3 millimeter long, and in each one there are around 1300 neurons which are responsible for sensing. So yes, so what we did today is we concluded the process of hearing by human ear. We have seen the actual sensing mechanisms in human ear, and we have also seen how the basilar membrane oscillations act as frequency separator as the first step towards frequency separation, and in that process we have theoretically divided basilar membrane into different sections. Each section is called a critical band, and the each critical band has different center frequency and different bandwidth. So we can stop here in today's lecture. We can continue we can continue on Google Meet in a short while from now.